Welcome to next class of generation of diversity of lymphocyte antigen receptor, though we are initial part is mostly the antibody or B cell receptor. Okay. So, in last lecture we ended up two segment V and J, they are heptamer, monomer, how they are coming, how this loop formed and it is deleted, or if the sequence is in the reverse direction or opposite orientation, then how this recombination happen. Okay. So, before studying this part or previous lecture this part, I will suggest you those who are not familiar with or do not remember much the recombination, you just go through the figures given in book about recombination. So, it will be very useful or helpful to understand, though I am trying to explain like what exactly happening, it is not that tough or critical. So, the thing is we are talking about diversity, how diversity is generated in receptors. Initial part VDJ, different number of or multiple number of VDJ segments, and then combination of different prime light chain and heavy chain makes the gives the two different component of diversity in antibody or B cell receptor. But what we are discussing in last lecture also in this lecture, how this VDJ join together, what are the proteins and how this, because it is important, because just that number, I mean that 2 points would have been essential, but there is something which will give another diversity. To understand that part, if we understand little bit detail, it is what I personally feel that to know little detail sometimes is good to generate the interest, like otherwise just to remember few lines are very tough. So, if you remember the whole picture what is happening, it will be very easy to remember the things. If you can remember the image, you can remember the figures, that can help you to remember the whole immunology. Okay. It is not like you have to read again and again and again. You just remember the figures, you understand the logic, and definitely you have to remember the correlation between the events, then you will see that immunology is not that tough. Okay. So, today's this lecture, next slide. So, do not be scared about seeing so many color and protein. What it is saying, that saying is basically the reaction that recombines VDJ different gene segments. It involves some very specific proteins, that lymphocyte specific protein which is not expressed anywhere else or any other cells. Not only that, it is also not expressed all the time in the lymphocyte. Just I told in the previous class, as soon as the recombination is done, many protein will switch up forever and so that no recombination happening. But some proteins are ubiquitous DNA modifying enzyme, because any recombination protein, there are various kinds of DNA repair system present in our cell, which helps us to save from mutations and any kind of deletion alterations in our chromosome. So, these protein or those proteins are also involved, which are some are common, which is present everywhere. Wherever there is a DNA break, they will act. Wherever there is a DNA break and repair, the say for example, the ligase which join two DNA fragment, that is common. So, there is a combination of lymphocyte specific proteins and ubiquitous protein, and here I will tell some the name of the protein, not all are that important from lymphocyte point of view, but you will see that many of them are very common. Okay. So, if you see, let me get the laser. So, if you see this B segment of this gene, and if this J segment 1 is again 23, it is again make it very small and thing. Let me go to little bigger version of that. So, this B segment where you can see this 23 here and here J is 12. So, now we will see how this J and B come together. If see first what happened, there is a protein called RAG, okay, RAG. It is actually the product of recombination activating gene. Recombination R activating A and gene G. recombination activating gene product, there is two different protein RAG1 and RAG2. This RAG1 and RAG2 make a complex. Okay, that I will show you just to show you how it make a complex. So, RAG1 and RAG2 make a complex, and this complex first bind to suppose this variable region B segment, rather V segment. Okay. So, this violet part is RAG1 and the blue circle is a RAG2. So, this is a heterodimer, and then they again make basically a tetramer, okay, 2 RAG1 and 2 RAG2. So, they first bind here, then what happened, another one binds here. So, what just in last class I told, one protein binds here, another protein binds here, they come together. So, what this going to happen? So, they will bring this B and J segment together, okay. And B and J segment when they come together, then there is endonuclease activity, that endonuclease will cut this 2 strand. So, one cut will be at V, one cut will be at V segment, another cut will be at J segment, and then they will be open. So, this cut, what you see this here, they cut this segment from the top part. So, there is a loop and the loop was cut, and this covalently closed DNA hairpin ends, that is very unique kind of thing that I will discuss later in this lecture only. So, this loop go out. Okay. Then what happened? This Ku70 and Ku80. What is this Ku? Ku, if you remember your recombination class, Ku is a heterodimer to Ku70 and Ku80, and which is a heterodimer. This Ku protein or Ku protein is doing what? It is making a ring like structure over the DNA, single stranded DNA, and it is happening both in case of this region also and this region also. I mean, in the bottom part also it is binding, upper part is also binding. So, this actually it is helping DNA recombination. This protein helps to DNA dependent, bring some DNA dependent protein kinase, which another protein is there, which we will see how it looks. So, if this is the Ku70 and Ku80 bind, Ku70 Ku80 bind, which is end, and this what we see here, this is a covalently closed DNA hairpin ends, that I will see tell you little detail later, and which is very interesting. And this then DNA PK, this violet big one, this DNA PK, the protein kinase come, then there is another protein Artemis come together, then this cutting and joining is coming going to happen. Then there is a TDT polymerase, which I will. So, this place, I mean this reaction part is very interesting, and I will tell you much more detail in molecular level what is exactly happening. Then DNA ligase and XRCC both are coming, which is again a DNA repair protein present. These Ku70, XRCC 4, DNA ligase, these are ubiquitous protein for cell. So, what as a result of this TDT function I will tell later. So, this cut Ku70 brings all this repairing non homologous end joining protein, double strand break repair protein. So, these are the protein of necessary for DNA recombination repair mechanism. So, if you want to study this detail, you go to any molecular cell biology book and or cellular molecular biology book, either way you read then you can find it there what exactly they are doing in recombination. There is nothing new for this purpose only. So, I am just mentioning the name. This is not, I do not want to go the detail of the recombination and what exactly going on in this recombination, but these proteins like all other regular recombination in cellular process, they are also taking part here. Okay. And ultimately, this makes a joining, and this join, if you see there, this is not only this red and yellow, right. This is not the red and yellow, there are few more colors are here. I will tell you why what these colors are. And in this case, the loop part, they come and the same complex happen, there will be cut, end joining, and the loop form and loop eliminated from the system, not in the same cycle. Next cycle of cell division, what will happen, that is outside the chromosome. So, next cell division cycle next cell cycle, that will not replicate. So, their loop will there till that cell survive. As soon as they divide, next cell or daughter cell, they will not be there anymore. Okay. This is how this RAG1 and RAG2 look like. This upper violet part is the RAG2 and this bottom part, the green and red, this is the RAG1. Why I am showing you, this is a crystal structure, just remember. So, what they have, the RAG1 has a gene dependent endonuclease activity. This is this enzyme active part or active part of the enzyme, and this part, this is bottom tail part, is NBD. NBD stands for nonamer binding domain. Nonamer means that same repeat. Okay. Nonamer binding domain. You will see the next slide will be little better to understand. So, this is same thing. So, different, that was a crystal structure, this is cartoon. So, cartoon is always better to understand. So, this part is a nonamer binding protein which makes a flexible hinge and they make a dimer here. This gene dependent nucleus domain and this is RAG2. What actually exactly happening? This nonamer binding site binds to this nonamer. This is the nonamer sequence. So, this is same say one DNA sequence, this is a variable region and this is the J segment, J region. So, V region, J region, both are heptamer. Okay. This region is heptamer and 2 heptamer, spaced by 1 is 23, another is 12, everything is I mean as before whatever we explained. So, this RAG1, this NBD domain interact with nonamer. So, if you go back and see the orientation or the arrangement of nonamer and heptamer, heptamer is just there is one heptamer very close to the segment that we need and then there is a nonamer. So, to nearest nonamer of V segment and J segment are brought together by this NBD domain. As soon as, so there is a common domain here which brings one sequence this side, another sequence this side. So, this is the V part and this is the J part. What will happen then, because 2 DNA is very close here. Okay. So, if there is this protein, this NBD actually brings them together. So, there this V segment is very close, J segment is very close, which is very close to the RAG. Okay. RAG1 is this gene dependent endonuclease. What it will do, it will cut here. You see the arrow, it will cut here. Okay. So, there is no heptamer sequence anymore. So, they will cut just after the heptamer and nonamer. So, in that region there is a specific site they cut and that cut will taken care by that Ku protein and all these things just we have seen. After Ku binding, we saw another protein that I told a few minutes back, the TDT. TDT is terminal deoxytransferase. Okay. Terminal deoxytransferase. What is the beauty of that? This enzyme can add nucleotide to the 3' end of DNA with a template independently. Normally DNA polymerase, what it is doing during DNA synthesis, you need a template and depending on that template and a primer, what you need, what the DNA polymerase is doing, if there is a they will add a T, if there is a G they will add a C, if there is a C they will add G, that you know the complementary DNA addition, that is how DNA synthesis happen. But terminal deoxytransferase, that particular enzyme has a unique capacity that they can add nucleotide randomly without any template. That means, if you give all possible nucleotide like 4 nucleotide like deoxy, ATP, deoxy, GTP, CTP and TTP random, they will add the nucleotide and there is how long they will add, as long it is here it is there is a limit, but there is a efficiency it can add nucleotide. If you give only A, it will continuously will add A, okay. But it will add at the double stranded DNA 3' end only. because DNA synthesis normally happen 5 prime to 3 prime synthesis. Okay. So, here is the magic how this TDT work. It is very, I mean, very wonderful mechanism. If you, I mean, I will go slow. Okay. So, this is, you see, this is a sequence which is 1, 2, 3, 4, 5, 6. This sequence, this is suppose here, I mean, previous slide we are talking about variable and V and J region. Here the example is given with D and J region, but it is true for any joining. Okay. Any two segment join, it is happening, Vj or Dj or Vd, similar thing is happening. So, in D segment, this is the last nucleotide suppose. So, D ends here, okay, and J ends here. What will happen? RAC complex bind and clips the recombination signal sequence. Okay, recombination signal, what is the recombination signal? You go and check, heptamer is here. Okay, this is, you will find. Now, if you, this RAC signal, RAC complex bind and cut, where it will cut? If you see this, let me get the pen first. So, RAC complex binds to the recombination signal and cleave that signal, and where it will cleave? You see this, it is cleaving just after T and C. Okay. It is cleaving just after T and C. In this case, it is after this TA. Okay. And normally what happen? If it cut, what is happening? If this is the DNA, if this is a DNA, if this is 5 prime and this is 3 prime and this is 5 prime, this is 5 prime and this is 3 prime, and what we know, all of you know, I am just repeating again, this is 5 prime is always there is a phosphate and 3 prime there is OH, and same way this is a phosphate and this is OH, right. So, there is a 3 prime OH and 5 prime phosphate. So, normally if there is any DNA, if this is cut, we can assume that this is the 3 prime on the upper hand side 3 prime, there is a OH here and there is a phosphate here. That is normally it happen if you cut, but this particular, and the same way it is here also, right. So, it will be phosphate and this will be OH. Okay. So, now what is happening, I will erase this part. Now, what is happening? Normally that will remain as free end, 5 prime phosphate and 3 prime hydroxyl end is remain free in a double stranded DNA. If you make a PCR or you just digest a DNA with a restriction enzyme, they will remain as free phosphate and free hydroxyl group. But here what is happening, which is very interesting, these phosphate and hydroxyl make a, these phosphate and hydroxyl make a link, okay, phosphodiester bond. So, open hydroxyl and phosphate will make a phosphodiester bond. So, what will happen? We will see a hairpin loop like structure. So, one DNA double stranded is there, but there will be a phosphodiester bond. Clear? So, that means it is a continuous thing, it is just a loop like structure. Then, so that is the beauty of RAG. Okay. So, endonuclease is cutting and making that hairpin loop at the end between the two strands. Then again it will cut here, you see the arrow, it will cut again here. So, what will happen? Then if you cut here the endonuclease, instead of only TC, it will get a sequence TCAG. So, this end is free. So, 2 GA here and AT here, it is extra, it was not there before, it was there in other strand, but not in the same strand. Now, if this DNA synthesized, if this DNA straightway synthesized, because this is 3 prime, okay. So, this DNA synthesized somehow, what we will see? We will see A, G, C and T, and same way if you see what you will see T A T A. So, this we will see, and if you complete, what will happen? It was originally this. If you just join this, if you just join this two end, if you just join this two end, there will be T C T A A G A T. It is supposed to be that only 4 nucleotide, but now due to this cut and hairpin loop, it is become 4 4 instead of 4, it will become 8. So, 4 extra nucleotide came in between this V and J junction, that was not there before. Okay. But it did not happen this way. It is not that straightforward that it will go and happen this, I mean synthesize this sequence. Okay. What happened? It will not synthesize that way. After making this sequence, so after making the hairpin and an internal cut, rag mediated cleavage generate P nucleotides. I will come what is that P, this P nucleotide. Then TDT come into picture, what I just told you. TDT is an enzyme which can add nucleotide randomly and template independent way to the 3' end of the DNA, 3' hydroxyl end of the DNA. So, what was there? It was there TCGA was there, okay. TCGA was there, and here it was TATA, and TDT is going to add the nucleotide randomly, and there is I mean nobody knows what nucleotide is going to come and how many number it will come. So, it will carry on like TCGA was then CTC, then it was AGCGAT, it is random, it is going to add. How long it will continue? So, these DNA are very close, they are together very close because all other protein kept them together. So, it will continue this way, another sequence is going to continue this way. What will happen as this addition of nucleotide is random? So, this random nucleotide also, if you continue, because there are only 4 nucleotide, there is not much variability in that. So, once they will get a complementarity. So, while this is synthesizing, they will make a complementarity, like so in upper strand CTC added and lower strand CGA is the last. So, what is happening this TA and CG? So, two DNA sequence of a single stranded DNA sequence was progressing like this, and sometimes they find the complementarity. So, as soon as they find complementarity, it becomes double stranded, right. It was like this growing and growing. As soon as they find complementarity, they become double stranded. As soon as it become double stranded, TDT cannot work, because terminal deoxytransferase can add nucleotide only to the single stranded. So, TDT stop working. Okay. So, what is happening and what happened? What happened? So, in this case, so this is 3' and this is also 3'. So, if there is any extra, that T will fall apart because they cannot match. Okay. So, then there is a DNA polymerase, that DNA polymerase which we are talking about, that DNA polymerase will continue to synthesize. So, what will happen is very simple. I am not going to draw here because it is already drawn here. So, if you see, it was up to only CTC, and then they pair between this AG and TC. This was paired during synthesis, then rest of the part will be synthesized this direction and this direction. So, they will fill up the gap by DNA polymerase, and after DNA polymerase fill up the gap, definitely there are ligase, that ligase will join this end. Okay, this is very common mechanism, every time it is happening. It is not very particular about this lymphocyte recombination inside the this T and B lymphocyte, but it is continuously it is happening in any cell, any time DNA repair, DNA breakage, excision repairs, deletion repair. So, there are so many varieties of DNA repair, variety of enzymes are there to repair the DNA. So, this machinery is not new, okay. But what is the interesting part of this? What is the result of this? This result is, it was only 2 before, okay. It was only 2 before when it was started with D and J. So, 2 nucleotide, now you see how many it become, it now it is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. That means, there are 11, so there are 2 to 4, now there are 13, that means 9 nucleotide was added in between. Okay. In that 9 nucleotide, if you see, in that 9 nucleotide, if you see that this part, the TCGA, 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 so in this part and in this TCGA, this sequence and TATA, this is palindromic. Palindrome, you know, palindrome normally we say the like if there is some word that which we can read either side, will same thing like madam, M-A-D-A-M, either side you read it is madam. There are many such word, but in DNA palindromic means that G-A-A-T-T-C, this is equal on one side, G-A-A-T-T-C. So, if you make, if you write G-A-A-T-T-C and the complement G-A-A-T-T-C. So, both direction, both the stand you did same G-A-A-T-T-C, G-A-A-T-T-C. So, that is 5 prime to 3 prime and 5 prime to 3 prime. So, if you see this, this is called palindrome in DNA. So, this sequence you will see that TCGA, TCGA. Okay. So, this part become palindromic, because it is eventually it should come, because if you see this how it happened, the origin is very clear. This palindromic part is called P nucleotide. In palindrome also there are 2 nucleotide extra. So, this P this is called palindrome, okay, because this is a palindromic sequence, both side it is a palindrome. But in between the 6th nucleotide, 5 nucleotide, that in between this GAGCG, which is added by this TDT, which is random. Okay. So, this randomness can make any combination of 5 or 6 nucleotide or 7 nucleotide, that is also not in control. So, it depends again, it is a random process. So, these 5 nucleotide which is randomly added in between D and J, this is called N nucleotide. So, now you imagine there are some fixed D sequence 1 to 23 and there are some J sequence, they can join, that gives you, suppose there are 10 D and 5 J. How many combination is possible if they are randomly, they any one of them can join 10 and 5, 50. But if this join make some nucleotide change which can contribute the number of amino acids is maybe 1, 2, 3, that number of amino acids can increase because of addition of the nucleotide. So, that variety is not anymore easily calculate, you can do like just by multiplication. Okay. So, this addition of P and N nucleotide or NP nucleotide in between the junction like DJ or VDJ. So, in case of heavy chain there will be 2 such cases, DJ one will happen and one VD recombination, 2 places it will add the N and P nucleotide. In case of light chain only in the CDR3, because the J segment and V segment is contributing the CDR3 region only. Okay. So, in light chain there will be only one place where this P N nucleotide addition is going to happen. In case of heavy chain there are 2 sites where N P nucleotide addition will happen, and that will very unlikely that each case or each time this addition is very similar or identical, okay, as it is completely normal, and this N and P nucleotide addition gives a huge amount of diversity, okay, huge amount of diversity, and which was this is the case, like if you see the germline theory and somatic theory, then what we see that definitely we are inheriting some genes or gene segment from our parents, like VDJ part is inherited from our parents, right. combination is ours, we do not know what is going to happen, and this N and P nucleotide addition is continuously happening every time the recombination is happened during the synthesis and the maturation. During the synthesis of B cell receptor or T cell receptor, that is totally random, and this NP nucleotide addition gives a huge diversity which was not known and not existed before, and this is the third reason of antibody diversity or the nucleotide lymphocyte receptor diversity. Okay. So, we will have very one more points left for that receptor diversity of B cell or B lymphocytes, and that will that time next lecture we will discuss that. Okay. See you then.